Hello everyone. Today we are going to discuss about respiration. Which the lecture will include the definition and explanation of respiration, phases of aerobic respiration, which includes glycolysis, Krebs cycle, electron transport chain, and we will study anaerobic respiration and fermentation. And lastly, we will discuss about the significance of respiration. So overall, the respiration basically is a biochemical process in which living cells oxidize organic substances like carbohydrates, amino acids to release energy in the form of ATP, and these reactions releases carbon dioxide and water in the as a byproduct, and most respiration is taking place in a presence of oxygen. And overall, this respiration is known as catabolic process, that is in which a complex food molecule is broken down into simpler ones, that by releasing energy in the form of ATP. So let's discuss about the difference between breathing and respiration. Breathing basically is a mechanical process that includes inhalation and exhalation, which the carbon dioxide from the cellular tissue of the body is released outside the body, where the from the atmosphere carbon dioxide is taken inside the body. Whereas respiration is a cellular process that occurs in all the living organisms, including plants, animals, fungi, microbes. All the living organisms uses cellular respiration to produce energy, and this cellular respiration is divided into two types. One is aerobic respiration, which actually requires oxygen, that occurs in presence of oxygen, and in which this aerobic respiration, the food molecule is completely oxidized. Complete oxidation of the food molecule takes place, and it also releases high amount of ATP. High ATP yield is occurring in case of aerobic respiration. Whereas in case of anaerobic respiration, what is happening? It is basically occurring in absence of oxygen, or without oxygen. This anaerobic respiration is taking place, where partial breakdown of the food molecule takes place. Food molecule is partially broken down to release very low amount of ATP energy. So overall, if we talk about the general equation of aerobic respiration, which includes the glucose is 6H12O6 plus 6 CO2, and it will release CO2 6 CO2 plus 6 H2O, and a total of 38 ATP. So one glucose molecule, if it is completely oxidized, it will release 38 ATP. So this cellular respiration basically includes three major reactions. One is glycolysis, that is glucose is broken down into 3 carbon molecule of pyruvate that releases ATP and NADH as a high energy molecule, and this pyruvate will go to mitochondrial matrix. It will go towards the Krebs cycle, and it will be completely broken down, and in the form we will get ATP, NADH, then FADH as a high energy molecule we will be getting from that Krebs cycle, and this high energy molecule will act as a electron donor for the electron transport chain that is occurring inside the inner membrane of mitochondrial inner membrane. Mitochondrial inner membrane contains different types of complex proteins that helps in the transfer of electrons, that we will discuss in later. So if we talk about the different phases of respiration, that includes glycolysis. First step of the aerobic respiration is glycolysis, which is common for both aerobic as well as anaerobic. The glycolysis is also known as Embden-Meyerhof pathway, as Embden and Meyerhof, the scientists who discovered this pathway, and based on their name, this glycolysis is also known as Embden-Meyerhof pathway. And the location for this glycolysis is cytoplasm. So this is the cytoplasm of the cell, is the place where glycolysis is taking place, where this one molecule of glucose is converted or broken down into 2 molecule of 3 carbon pyruvate. Then if it is the simplified reaction, if we talk about glucose first converted into glucose 6 phosphate, then glucose will become 6 phosphate to fructose 6 phosphate, fructose 6 phosphate to fructose 1,6 bis phosphate, then it will be clipped out to form 3 carbon sugar molecule, and it will be further oxidized to produce pyruvate. So in this glycolysis, 4 ATP is produced, and in the initial reaction we use 2 ATP, So overall net gain from the glycolysis will be 2 ATP, and we will be additionally we will be getting 2 NADH molecule, that will be an equivalent to 6 ATP. That now if we discuss the location where these different types of stages of aerobic respiration is taking place, the glycolysis, the site of glycolysis is cytoplasm, and the Krebs cycle is taking place inside the matrix, inside this mitochondrial matrix, where this Krebs cycle is taking place. Krebs cycle is taking place, and this inner membrane, mitochondrial inner membrane, contains this complex proteins that helps in the electron transport chain. And mitochondria, it has own DNA, it has its own circular DNA, it has its own ribosomes also. So it has this capacity to produce all the proteins required for its functionality. So if we talk about this pyruvate, then goes to mitochondrial matrix, then it will combine with acetyl CoA, then it will produce acetyl CoA. In this process, one CO2 is released, and we will be getting one NADH. So as we have produced two pyruvate from glycolysis, so from this link reaction we will be getting 2 NADH, and overall 2 carbon dioxide will be also be reduced. So 2 acetyl CoA, 2 NADH, and 2 carbon dioxide is released. So we are getting 2 NADH molecule as a high energy currency molecule that we will be using in the electron transport chain. So if we talk about Krebs cycle, it is also known as citric acid cycle, or sometimes called tricarboxylic acid cycle, or TCA cycle. So the location for Krebs cycle is mitochondrial matrix. So we need to remember what are the different phases of this aerobic respiration, which is the location where it is taking place. Basically, sometimes questions ask that what is the location for the Krebs cycle, or what is the location for the link reaction as such. So in this Krebs cycle, the acetyl coenzyme A combined with oxaloacetic acid to produce citrate. So as the citrate is the first stable product, that's why we are calling it as a citric acid cycle also. In this reaction, a series of different enzymatic reactions leads to production or release of CO2 and production of NADH, FADH2, and also ATP. So overall product of this Krebs cycle, if we talk about, we are getting 6 NADH, 2 FADH2, 2 ATP, and 4 CO2 is released. So this is basically the Krebs cycle. Krebs cycle that includes like acetyl CoA combined will by oxaloacetic acid bind with oxaloacetic acid to produce citrate to isocitrate to alpha ketoglutarate. In this process, one CO2 is released and one NADH is produced. Then alpha ketoglutarate to succinyl coenzyme A. In this case, another CO2 is released and NADH is formed. Then succinyl coenzyme A is then formed succinate. In this process, coenzyme A is released and one GTP is formed, and GTP is basically equivalent to ATP. Then succinate forms fumarate. In this process, FADH2 is produced, and fumarate then converts into malate to oxaloacetic acid. In this process also we are getting one NADH. So overall there are 3 NADH and 1 FADH2. As we have produced 2 pyruvate to 2 oxaloacetic acid, so this cycle will run twice and it will produce a total of 6 NADH, then 2 FADH2, and also 2 GTP, that is equivalent to ATP. This is in general the electron transport chain we are talking about. There are 4 major complex protein. Complex 1 is NADH reductase, complex 2 is cytochrome reductase, complex 3 is cytochrome oxidase. This cytochrome reductase is this 2 is cytochrome reductase, complex 2 and complex 3, where this NADH reductase, where NADH will be converted into NAD plus. In return it will release 2 hydrogen and 2 electron. This electron will transfer through several electron transport carriers towards the complex 4, where this cytochrome reductase also reduces FAD from FADH2 to FAD. In return the succinate to fumarate is converted, and in that case also 2 electrons is released, and this electron energy is used for the transfer of photons from the matrix to the intermembrane space, which this area will have high amount of photon gradient will be produced, and this photon can only come out from this molecule, that is F0 F1 particle. So this is the reason why it's a motor protein that uses this energy from the photon to create a phosphate bond between ADP plus Pi. This will club together to form ATP. So this is the final reaction where the ATP for majority of the ATP is formed, and this electron, the terminal electron, is carried by the oxygen. So this is the oxygen is the terminal electron acceptor for this electron transport chain. So this is in general the electron transport chain. This high photon gradient will come out from this ATP synthase, and from ADP plus Pi will combine to form ATP. So overall, if we talk about the reaction of the terminal electron transport chain, where it is occurring inside the mitochondria, inner mitochondrial membrane, where NADH FADH2 donates electron to the electron carriers like cytochromes, ubiquinone, etc. So the flow of electron is downhill, and final electron acceptor is oxygen, and this photon gradient that is built inside the intermembrane space that drives the ATP synthesis. Basically it forming a chemo-osmotic synthesis of ATP, and it is also known as phosphorylation of ATP. So overall we are getting each NADH is equivalent to 3 ATP, FADH2 is equivalent to 2 ATP. So in general, from that we are getting total 34 ATP from electron transport chain, and we have got 2 ATP from glycolysis and 2 ATP from Krebs cycle. Krebs cycle, or in general as well, there were two pyruvate, so 2 into 2, there are 4. So in total 2 ATP, 2 to 4, and this 34. In general we are getting total of 38 ATP, 36 to 38 ATP we are getting from oxidizing one molecule of glucose. So glycolysis produced 2 ATP, link reaction produced 2 NADH, Krebs cycle produced 2 ATP, 6 NADH, 2 FADH2, and electron transport chain produces a total of 34. So in general we are getting this 34 to 36 and 2 38. In total 38 ATP we are getting from one glucose molecule. Now if we talk about the anaerobic respiration, as we all know that it occurs without the involvement of oxygen. It does not require oxygen. So where the incomplete oxidation of the food component taking place, and where the anaerobic respiration taking place, anaerobic respiration is taking place inside the cytoplasm. So end product of this anaerobic respiration can be of lactic acid or ethanol, and CO2 is released in the form. So overall we are getting only 2 ATP molecule per glucose molecule. We are getting only, that's why we are calling it as a very low energy yield or ATP yield from this anaerobic respiration. So lactic acid basically synthesized inside the human muscle cell during vigorous exercise. If we do running or vigorous exercise, during that our muscle cells do not get enough amount of oxygen. So in absence of oxygen, muscles cells uses this anaerobic respiration process to supply energy to the cell. So in that case it produces lactic acid. The pyruvate is converted into lactic acid. So pyruvate will be converted into lactic acid, thereby we are getting 2 ATP. But this lactic acid, when it gets deposited or stored inside the muscle cell, it creates fatigue inside the muscle, that creates pain or muscle cramp. Whereas in case of microbes, the yeast, this pyruvate is converted into ethanol. Pyruvate is converted into ethanol, which has several industrial or application also. So next, if we talk about fermentation, the fermentation is also an anaerobic process where pyruvate is converted to ethanol and CO2 is released. The fermentation is carried out by yeast, which is very useful for the production of wine, beer, bread, etc. Now if we talk about lactic acid fermentation, this is where the pyruvate is converted into lactic acid. This occurs in the muscle, or also certain bacteria known as lactic acid bacteria, that or like lactobacillus. This lactobacillus also uses this anaerobic respiration or lactic acid synthesis mechanism to produce lactic acid, which we can be used in the curd, cheese, yogurt. These are the food components that we use daily. So this is also used by the lactic acid bacteria. So overall, fermentation also very inefficient. It only uses release 2 ATP and does not require any mitochondria. And although it has important features for food and beverage industry, this fermentation has very significant importance in the food and beverage industry. If we talk about overall significance, why respiration is significant for us, so it provides ATP production, so energy currency of the cell. So ATP used for different processes like biosynthesis, transport, contraction, nerve impulse, all those activities inside the living cell requires ATP. So this ATP is coming from the respiration. Metabolic intermediates like Krebs cycle intermediates used for synthesis of amino acids, and nucleotides and fatty acids. The intermediate components that are generated during Krebs cycle are used for the synthesis of different amino acids, nucleotides and fatty acids. Like physiological functions in plants drives growth, germination, maintenance, respiration. All those things are supported by this respiration. In animals also it supports movement, thermoregulation, brain function. All those activities are regulated by respiration, as this activity requires energy, and energy is coming from the respiration. Like industrial application, like fermentation has huge implication in alcohol production, bread, curd, cheese, antibiotics. All those things have significant implication of this fermentation. Also the adaptive and survival, certain microorganisms are facultative anaerobics, means they can survive in absence of oxygen or in presence of oxygen also. So like yeast and certain bacteria, they can survive without oxygen. In presence of oxygen they uses aerobic respiration process, whereas in absence of oxygen they will use anaerobic respiration. Medical significance, this is mitochondrial dysfunction is related to several type of diseases, and anaerobic respiration like lactic acid leads to hypoxia or respiratory quotient used for clinical diagnosis also. The respiratory quotient basically means it's the amount of CO2 released and amount of O2 consumed. Like for example, if in case of glucose or carbohydrate, if we use for 6 carbon glucose, you need 6 O2, and it also releases 6 CO2. So this respiratory quotient will be 1. Whereas in case of fat, it has very high oxygen demand, but it releases very low amount of carbon dioxide. That's why fat has 0.7 RQ, whereas protein have 0.9 RQ, 0.8.52 or 9 around RQ. So if we discuss little bit about the comparison of aerobic respiration and anaerobic respiration, so oxygen requirement in aerobic respiration it is required, in anaerobic it is not required. Location, and aerobic respiration taking place cytoplasm, part of cytoplasm and part in the mitochondria, whereas anaerobic respiration is taking place only inside the cytoplasm. End product we are getting CO2, because the glucose molecule is completely oxidized and CO2 is released and H2O is also released. Whereas in case of anaerobic respiration, this pyruvate is converted into lactic acid or ethanol, which, if we talk about the energy efficiency, this 36 to 38 ATP per glucose molecule we are getting, and it's a high energy efficiency. Aerobic respiration is high energy efficient mechanism, whereas anaerobic respiration we are getting only 2 ATP per glucose, and it is very low energy efficient molecule reaction. In organism, if we talk about, most plants, animals uses an aerobic mode of respiration, because the plants and animals require oxygen for the respiration. But certain microorganisms like yeast and like bacteria, or also human muscle cell under different stress, uses this anaerobic respiration process to provide energy to the cell functional. So thank you for listening. So follow for similar lecture. We will continue with the plant nutrition and plant transport, and subsequent chapters we will discuss in later lecture. So kindly follow, and also comment your queries in the comment box, so that I will try to resolve them as much as possible. So thank you for